Okay, so thanks everybody. The work is, it's quite a few people involved. As you'll see, there are a few authors there. So I'm going to talk about three things today. I'm going to talk about, firstly, richness and abundance patterns across the altitudinal gradient. Then I'll say a little bit about functional diversity across the gradient. And then to end off, I'll talk a little bit about climate change and the future, where we should be heading with this. So in terms of altitudinal gradient studies, diversity changes across an environmental gradient. They're useful for allowing us to understand which environmental factors influence diversity patterns. We can also, from them, we can understand the relative roles of biotic and abiotic factors in structuring assemblages. And we can also document natural fluctuations in abundance of particular species. And then also over the long term, we can look at responses to climate change. So the Sani Pass gradient, where we've been working, we started working there in 2003. We've only been sampling across this more extensive gradient from 2006. The sampling is from 900 meters up to 3,000 meters. We have 8 different sites spread across that gradient, and they're 300 vertical meters apart. We sample in 2 periods, one in the wet season in January and in the dry season in September. And sampling so far, we've done 9 years of sampling in January and September. We use pitfall trapping because it's simple, it's low tech, and it's repeatable. And then why ants? Well, they respond to environmental change more quickly than long-lived species. They're relatively easily sampled. Sampling is repeatable. Taxonomy is fairly good. And we can actually study whole assemblages, which is quite important. They also have a wide range of functions: predators, important mutualists, herbivores. There are also some important invasive species. Luckily, no invasive species that we've recorded across this gradient. They also play an important role in ecosystem function. So now, the first part of what I'm going to talk about today is the species richness patterns. So there's generally a decrease in richness with an increase in altitude. And several studies have reported a hump-shaped species richness relationship, with the explanatory variables for this being temperature, precipitation, area, and what we call geometric constraints. A few studies have actually considered change in diversity across gradients over a long period of time. So they've just focused on a couple of years of sampling, and that's it. So where our study is slightly different is that we have many different years of sampling. And one of the other things is that we can get valuable insights from looking into drivers by considering both time and also season. So very few studies have considered gradients in different seasons. So I'll talk about 2 aspects. I'll talk about species density, which is, if you don't know the difference between species density and species richness, think of it in terms of species richness, and then also patterns of abundance across several years and in 2 different seasons. So this is the sampling protocol we use, as I said, starting at 900 meters, going up to 3,000 meters. We've got 4 replicates per site, each replicate separated by 300 meters, and at each one we have 10 pitfall traps in 2 rows separated by 10 meters. We use that same size of pitfall trap that was shown earlier on, and we also use these rain covers to prevent the traps from being flooded by rain. We also have temperature monitoring. We monitor the temperature just below the soil surface. We're recording at each of those sites 2 different replicates every hour. So we've got a fairly good temperature record. So in terms of results, well, we've sampled 440 communities. That's 8 sites, 4 replicates, 2 seasons, and 7 years. So we have, based on that, we've got over 60,000 individuals. That's 90 species. Thanks, I'll try and use the pointer now. My hand also works. So maybe in this case, being a bit shorter, it doesn't work so well. So the red lines are the wet season, and the blue lines are the dry season. Okay, so these are the different years, and that's altitude over there, going from low to high altitude. So a clear hump-shaped relationship. This is a summary of all years. So there's some variation between years, and that is in terms of the species density. So clear hump shape, and differences between seasons. So more species in the wet season than in the dry season. In terms of abundance, the relationship is a lot more noisy. There's also a difference between the two seasons, and there's also a difference across the years. Okay, so what we're getting in terms of the diversity patterns, we're getting mid-elevational peaks in species density. The abundance patterns are slightly more complicated, but temperature seems to play an important role in explaining the patterns of species density and abundance. There's also greater seasonal variation at low elevations. So less seasonal variation at high elevations, more seasonal variation at low elevations. So if we look at some of the predictors of these patterns, we fitted some models to figure out which were the best explanatory variables. We get, for species density, the best models we find included mean temperature being quite a strong predictor, and area, amount of area at a particular altitude, being a weak predictor. So the interpretation is that low temperatures are likely to limit foraging, and we find fewer species active during lower temperatures. In terms of abundance, temperature and standard deviation in temperature were the best predictors. We know that ants are known to be thermophilic, so they prefer warm periods, rather warm temperatures. So if we look at the role of temperature, temperature is an important factor driving ant diversity. We find more individuals and more species in hotter parts of the gradient and during warmer periods. So temperature seems to be an important factor driving the diversity patterns. An important point to highlight here is that mechanisms that don't account for seasonal effects can't be primary drivers. So area doesn't change across season, but yet the patterns change. So therefore, it's not possible that area is actually the mechanism driving the pattern. So it's clear to us that temperature has quite an important influence in driving these richness patterns. And so we need to think about temperature, and especially in terms of climate change implications. If temperature is driving the patterns, then climate change is likely to have a large impact. Okay, so now moving on to the functional diversity component. This is the second of the three components I'm talking about. So you can see these are not ants, these are birds. And you can see that they have different beaks, and you can infer that they do different things based on those beak shapes. So that's the idea behind a functional diversity, looking at the different functions that things perform. So we asked the question, well, what is the role of species in the assemblage? What does it do? Which ordinary species diversity doesn't tell us. So you can infer function from morphometric measurements of ants. You can look at body length, you can look at eye position, leg length, mandible length. These things can tell you a bit about what the species do. And so if we look at some of the measurements that can be taken, things like body length, you can look at relative leg length. So if you take those two measurements, you add them together. You can also look at relative mandible size and also eye position. These can be informative. So if we look at these in terms of the functional traits, you get predatory specialists, which have relatively large mandibles, laterally positioned eyes, and relatively short legs. So those, the predatory specialists fit into that category. Then those with generalized traits, relatively short mandibles, dorsally positioned eyes, and relatively long legs. And then if we look at species that occupy dense, complex habitats, these generally have small bodies, relatively short mandibles, and short legs. And species that occupy open, simple habitats tend to have larger bodies, relatively long mandibles, and relatively long legs. So if we look at the functional space, so this is an ordination where each dot represents a species, and these are the different variables: mandible length, that's Weber's length, the body size measurement, that's a leg length, and that's an eye position. So what you'll have to do is believe me that what I'm saying is the truth. So this is a community at 900 meters. So if you put all of the species in the 900 meter community, so that's the lowest altitude, spans that amount of the functional space. The community at higher elevation, that's at 3,000 meters, occupies a much smaller functional space. And what I should have actually given you first was what these axes mean. So this axis here we interpret to represent some measure of habitat selection. So these would be the dense complex habitats. These would be the open and simple habitats. So that's how we interpret that axis. The next axis here, we look at being the predatory specialists and those with generalized traits. So we can see that certainly the ones that are dropping out towards the higher elevations, you've got a much more generalized type of species that occurs there. So I think what's exciting for me is that you can see that the functions change across the altitudinal gradient, which I think holds quite a lot of promise for us understanding how these systems actually function. So just to summarize, a smaller part of the trait space occupied at higher altitudes. The high altitudes are characterized by the generalists. The specialist predators only occur at low altitudes. Now, the third part of what I want to say today is about climate change and altitudinal gradients. So this is really what we're hoping to get at over the longer term. We've been at this for this coming year will be the 10th year. Hopefully we can keep this up in the long, over the long term. What we expect to see with climate warming would be shifts in elevational ranges. So if this was the current range, you would tend to see new populations of that species appearing at higher elevations as the temperature increases. These areas that were previously too cold now become suitable. These areas that were suitable now become too hot for the species, and they become locally extinct over there. So this is what we'd expect, altitudinal range shifts. But we need to think that obviously these things don't happen in isolation. Not all species are going to shift at the same rate. There are going to be new competitors, so species will encounter new competitors. They'll also potentially encounter new predators, and spiders could be some of those predators. You also get changes in prey abundance. So this species, this Plectroctina mandibularis, is a specialized predator on millipedes. There'll be changes to mutualisms. Here's some ants tending aphids. There'll also be biological invasions. This is the Argentine ant. Hopefully we don't see that arriving. And then also obviously important changes to ecosystem function and to ecosystem services. This species dispersing a seed. So in terms of climate change studies, altitudinal gradients are ideal for these. You can get a good understanding of what's actually happening across the whole gradient. It's best to study whole assemblages. You want to sample all of the species in the assemblage, and you would also best do it with repeated sampling and standardized methods. And obviously long-term studies are the ideal here. You also want to minimize confounding factors like land use change. And I think we've got a long-term study. We've got the beginnings of a long-term study. So in terms of ongoing future work, I've got a good PhD student, Tom Bishop. He's currently examining beta diversity across the gradient, has just written a paper on functional diversity and taxonomic diversity. I showed you some of the results of that. He's also interested in understanding food preferences of the species. also activity periods, when they're active day or night or different times during the day. He's also done some work on the physiology of these species. So he's got a paper published in Journal of Biogeography. There's another one in review, and hopefully there'll be many more to come. So the conclusion, altitudinal gradients useful for understanding ecosystem function. Long-term monitoring in different seasons is valuable for understanding climate change responses. Acknowledgements, many people have been involved. CIB, Centre for Invasion Biology, is one of the key funders of this, and it wouldn't be possible without their contribution. There also Masda Wildlife Fund, Chantal Ferreira is an absolute gem because she can identify ants. She has a lot of patience. We also rely on a lot of students that have helped in the sampling. Thank you very much.